Today in this presentation, I will going to talk about nanotechnology in health. I want to I want to talk about the other topics like what is the nanotechnology and the size of the nanotechnology and technology in health, like the using in chemotherapy and like with fighting with a cancer. I'm gonna talk about this. The scientist Richard Phillips Feynman is the first to state that nanoscale studies can be done. He was born in America in 1918. I want to talk about the what is the nanotechnology, like designing and using structures between 1 and 100 nanometers to serve a specific purpose is called nanotechnology. Now I'm going to talk about the size of the nanotechnology to show you how small it is. Let's consider the nanoscale. When we divide 1 meter into 1000, we get 1 millimeter. When we divide this into 1000 again, we go down to 1 micron. Microbes of this size and can only be seen with a microscope. When we divide this into 1000, we come to the nanometer finally. Nanotechnology in health. Nanotechnology creates the opportunity to interfere with living systems at molecular levels. It is possible to develop many diagnostic and treatment methods by producing tools that can interact with living organisms. The use of nanotechnology in healthcare. Nanotechnology is also used a lot of in the field of health. Nanotechnology is used in the diagnosis, treatment, and most areas of the disease. For example, it is used in the diagnosis and treatment of diseases that cause major problems such as cancer, tumor, and many more. Now I am in my main topic, like nanotechnology in chemotherapy. There is ongoing research on the use of nanoparticles in chemotherapy. Chemotherapy, which has been used for years to treat cancer, can cause serious damage to the human body. Using nanoparticles, for example, it may be possible to destroy cancer tumors with minimal damage, to healthy tissue and without the serious side effects often caused by chemotherapy treatments. I want to talk about a survey of nanoparticles in chemotherapy. One treatment under development involves targeted chemotherapy that delivers a tumor-killing agent called tumor necrosis factor alpha, like TNF, to cancer tumors. TNF is attached to a gold nanoparticle along with thiol derivatase, polyethylene glycol, which hides the TNF-bearing nanoparticle from the immune system. This allows the nanoparticle to flow through the bloodstream without being attacked. The company developing this targeted chemotherapy method to deliver TNF and other chemotherapy drugs to cancer tumors is called Sliman. Another targeted chemotherapy treatment under development uses a nanoparticle called CRLX 101. The company developing this targeted chemotherapy method is called Cerulean Pharma. Crystal Therapeutics is conducting phase 1 clinical trials using nanoparticles called 3-pack to deliver a drug called docetaxel to tumors. Researchers at MIT are developing nanoparticles designed to pass through the brain barrier and target tumors of a type of brain cancer called glioblastoma, delivering two chemotherapy drugs to the tumor. Researchers at the University of Toronto have demonstrated the use of manganese dioxide nanoparticles designed to concentrate in a tumor and generate oxygen can increase the effectiveness of the chemotherapy drug doxorubicin. Researchers at UCLA have demonstrated the use of mesoporous silica nanoparticles to deliver the chemotherapy drug irradiation to pancreatic cancer tumors. Testing on mice indicates that this method reduced the toxicity of the chemotherapy. Researchers at the University of Georgia are working on a method to fight prostate cancer. They are using nanoparticles to deliver a molecule called IPA3 to the cancer cells. In laboratory mice studies, the IPA3 appears to reduce the growth of prostate cancer cells. Researchers at the University of Texas Southwestern Medical Center have used nanoparticles called dendrimers to deliver nucleic acids to suppress nucleic acids that suppress tumors to liver cancer tumors. The researchers have demonstrated in lab tests that this method can reduce tumor growth in mice. Researchers at the University of Leicester and three other universities are developing synthetic polymer nanoparticles or nanomips, which may result in improved delivery of chemotherapy drugs to cancer cells. Researchers are developing graphene strips to deliver different drugs to specific regions of cancer cells. When the graphene strip reaches the cancer cell, one drug separates from the graphene and attacks the cell membrane, while the graphene strip enters the cell and delivers the second drug to the cell nucleus. Researchers at MIT are developing nanoparticles that carry precise ratios of three different drugs. They are testing the effectiveness of this approach on ovarian cancer cells. Researchers at the Johannes Gutenberg University Mainz have developed a method to attach antibodies to nanocapsules that they believe builds more stable nanocapsules, which will improve the ability of the nanocapsule to deliver drugs to cancer cells. Researchers at UCLA are investigating a method to fight pancreatic cancer using two different nanoparticles. The first nanoparticle removes material on the exterior of the cancer cells that block the entry of chemotherapy drugs. The second nanoparticle carries the chemotherapy drug. Testing this method on laboratory mice showed significantly faster shrinkage of the tumors than other methods. Researchers are testing the use of chemotherapy drugs attached to nanodiamonds to treat brain tumors. The nanodiamond chemotherapy drug combination stays in the tumor longer than the chemotherapy drug by itself, which should increase the effectiveness. Researchers are also testing the use of chemotherapy drugs attached to nanodiamonds to treat leukemia. It turns out that leukemia cancer cells can pump chemotherapy drugs out of the cancer cell. Limiting the effectiveness of the drug, the cancer cell can not pump the nanodiamond out, So attaching the drug molecules to nanodiamonds results in the drug staying in the cancer cell longer. Researchers are connecting different DNA strands together into a structure they call a nanotrain. They have demonstrated in lab studies that these nanotrains are effective in delivering chemotherapy drugs to cancer cells, and that by using different DNA strands, they can customize which type of cancer cells our nanotrains target. Researchers are developing a nanoparticle that both uh, delivers delivers a chemotherapy drug and stimulates the immune system to attack cancer cells. They have tested the method on mice with positive results. A method being developed to fight bladder cancer uses nanoparticles called micelles to deliver a chemotherapy drug called paclitaxel to bladder cancer cells. An improved way to shield nanoparticles delivering chemotherapy drugs from the immune system has been developed by forming nanoparticles from the membranes of red blood cells. Researchers have demonstrated a method of delivering a protein to cancer cells that destroys the cancer cells. They use a polymer nanoshell to deliver the protein into the cancer cells. When the protein accumulates in the nucleus of the cancer cell, the protein causes the cancer cell to self-destruct. Using polymer nanoparticles to deliver a molecule called GSI 124 to cancer tumors, this molecule degrades the ability of the cancer cells to suppress the immune system, possibly slowing the growth of cancer tumors. Another technique delivers chemotherapy drugs to cancer cells and also applies heat to the cell. Researchers are using gold nanorods to which DNA strands are attached. The DNA strands act as a scaffold, holding together the nanorod and the chemotherapy drug. When infrared light illuminates the cancer tumor, the golden nanorod absorbs the infrared light, turning it into heat. The heat both releases the chemotherapy drug and helps destroy the cancer cells. Researchers have demonstrated a nanoparticle that kills lymphoma cancer cells. They use a nanoparticle which looks like HDL cholesterol but with a gold nanoparticle at its core. When this nanoparticle attaches to a lymphoma cell, it blocks the cancer cell from attaching to real HLD cholesterol, starving the cancer cell. A method being developed to fight skin cancer uses gold nanoparticles to which RNA molecules are attached. The nanoparticles are in an ointment that is applied to the skin. The nanoparticles penetrate the skin and the RNA attached to a cancer-related gene, stopping the gene from generating proteins that are used in the growth of skin cancer tumors. My presentation is ends in here, and thank you for listening to me, and have a nice day.